Welcome to the PubMed Daily Review for April 11th, 2026. Today we scanned 3,422 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, a living therapeutic platform for localized in situ modulation of macrophage pyroptosis ameliorates GVHD while preserving GVL by Zhuo Wan and Weiwei Qin from Hematology, Tongdu Hospital, 4th Military Medical University, Xi'an, 710038 China. Let's break down its key contributions. Graft versus host disease, or GVHD, is a serious complication after stem cell transplants, often affecting the gut. We now understand that a specific type of cell death in immune cells called pyroptosis, particularly in macrophages, plays a big role in causing GVHD. The challenge is that existing treatments for GVHD can weaken the body's ability to fight cancer, so we need a more targeted approach. To tackle this, researchers engineered a probiotic bacterium, E. coli Nissl 1917, to produce special nanoparticles called outer membrane vesicles. These vesicles were loaded with a peptide called big LEN, designed to specifically block the NLRP3 inflammasome, a key trigger for pyroptosis. They tested this engineered probiotic system in mouse models of GVHD and in lab experiments with macrophages, also analyzing human data to confirm their findings. The results were very promising. The engineered OMV therapy significantly reduced the damage caused by GVHD in the mouse models and improved survival rates. Importantly, this localized treatment did not interfere with the beneficial graft versus leukemia effect, meaning the transplanted cells could still fight the cancer. The big LEN peptide showed a strong binding affinity to its target, NLRP3, confirming its potential mechanism of action. This study demonstrates a powerful new approach using engineered probiotics to deliver targeted therapies directly to the site of inflammation. By specifically modulating macrophage pyroptosis, they were able to effectively treat GVHD without compromising anti-cancer immunity. This innovative platform could pave the way for new treatments for various inflammatory conditions in the gut. Up next, our second paper out of 5, Accelerating Personalized Medicine, Miniaturized Patient-Derived Organoid Drug Screening for Predicting Cancer Treatment Responses and Beyond by Yasmin Aboulela and Sylvia F. BOJ from Organoids BV, Hub, Utrecht, the Netherlands, now as part of the life science business of Merck KJ, Darmstadt, Germany. Let's break down its key contributions. Predicting how a patient's cancer will respond to different treatments is crucial for personalized medicine. Patient-derived organoids, or PDOs, offer a way to test drugs on a patient's own tumor cells outside the body. While promising, the current methods are slow and require a lot of precious sample, making it hard to use them quickly in the clinic. This research sought to overcome these limitations by developing a faster, more efficient screening method. The researchers focused on optimizing an automated system called the Yamaha Cell Handler, or YCH, to handle patient-derived organoids. They developed a miniaturized assay using 384 well plates and tested various parameters like seeding density and extracellular matrix concentration. The goal was to significantly reduce the number of organoids needed per test. This automated platform was then validated by comparing its drug sensitivity predictions against traditional methods and actual patient responses. The automated YCH system achieved an impressive 80 to 100% transfer efficiency for PDOs, allowing for the use of just 5 to 10 organoids per well. This represents a significant reduction in sample needs, potentially 30 to 50-fold less than conventional methods. Crucially, the drug responses observed in the miniaturized organoid assay closely matched the actual outcome seen in patients, validating its predictive power. This study presents a significant advancement in making organoid-based drug screening more practical for clinical use. By automating and miniaturizing the process, it drastically reduces the time and sample needed to predict treatment responses. This could lead to faster, more tailored treatment decisions for cancer patients. While promising, further studies are needed to fully integrate this technology into routine clinical practice. Up next, our third paper out of 5, Molnupiravir or nirmatrelvir-ritonavir plus usual care versus usual care alone in patients admitted to hospital with COVID-19. Recovery, a randomized, controlled, open-label, platform trial by Peter W. Horby and Martin J. Landry. Let's break down its key contributions. We know that oral antivirals can be effective for treating COVID-19 in the outpatient setting. But what about patients who are sick enough to be admitted to the hospital? Data for these individuals is limited. This study aimed to answer whether adding Molnupiravir or nirmatrelvir-ritonavir to usual care could improve outcomes for hospitalized COVID-19 patients. This was a randomized, controlled, open-label trial, part of the larger recovery platform. Patients were assigned to receive either Molnupiravir or nirmatrelvir-ritonavir alongside their usual care, or just usual care alone. The main thing they looked at was whether patients survived for 28 days. The study took place across multiple hospitals in the UK. In the Molnupiravir group, 17% of patients died within 28 days, which was the same as the usual care group, with a hazard ratio of 0.93. For nirmatrelvir-ritonavir, 19% died in both groups, with a hazard ratio of 1.02. There were no significant differences observed in how long patients stayed in the hospital or if they needed invasive ventilation. Essentially, these antivirals didn't show a clear benefit in this hospitalized population. So what's the takeaway here? Adding Molnupiravir or nirmatrelvir-ritonavir to usual care didn't lead to better outcomes for hospitalized COVID-19 patients in this study. However, recruitment was low, meaning we can't completely rule out a benefit, particularly for nirmatrelvir-ritonavir. Future studies might be needed to see if certain patient groups could still benefit from these treatments. Up next, our fourth paper out of 5, a generalizable foundation model for analysis of human brain MRI by Divyanshu Tak and Benjamin H. came from Intelligence in Medicine, AIM, program, Mass General Brigham, Boston, Massachusetts, USA. Let's break down its key contributions. Analyzing brain MRIs holds immense potential for revolutionizing how we diagnose and treat neurological conditions. But the field has been held back by a lack of sufficient training data and models that are too specialized, failing to perform well across different patient groups or tasks. Foundation models, leveraging self-supervised learning, are emerging as a powerful paradigm to tackle these challenges by learning generalizable features from unlabeled data. To address these issues, the researchers developed Brainiac, a foundation model specifically for brain MRI analysis. Brainiac was trained using a self-supervised contrastive learning approach on over 32,000 unlabeled brain MRI scans. This method allows the model to learn robust, generalizable features without requiring task-specific labels. The core idea is to create a versatile model that can be easily adapted for various downstream applications, from classifying MRI sequences to predicting survival. The results were impressive. Brainiac demonstrated superior performance across all evaluated downstream tasks, including mutation prediction and tumor segmentation, especially when training data was scarce. It significantly outperformed traditional supervised training and other pre-trained models, even in challenging few-shot learning settings where only 1 or 5 examples were available per class. This adaptability makes Brainiac a powerful tool for real-world clinical applications where labeled data is often limited. In conclusion, Brainiac represents a significant advancement in AI for brain MRI. Its generalizability and exceptional performance in low data conditions pave the way for more robust and widely applicable AI tools in neurology. This foundation model could accelerate the discovery of new biomarkers and significantly speed up the translation of AI research into tangible clinical benefits for patients. Up next, our fifth paper out of 5. CD24 as an innate immune checkpoint in solid tumors. Biology, Biomarker Stratification, and Therapeutic Translation by Weiwei Zhou and Wenxiu Ma from Hunan Provincial People's Clinical Medical School of Zhengzhou University, Zhengzhou, Hunan, China. Let's break down its key contributions. Solid tumors continue to be a major challenge in cancer treatment, largely due to their ability to suppress the immune system. While we've made great strides targeting adaptive immunity, like with PD-1 blockers, many patients don't respond or develop resistance. This has led researchers to focus on the innate immune system and its role in tumor evasion. One molecule emerging as a critical player is CD24, which acts as an innate immune checkpoint by dampening the anti-tumor activity of macrophages. This paper is a comprehensive review, synthesizing current knowledge from various sources, including lab experiments, animal studies, and analyses of human tumors. The authors meticulously reviewed existing literature to understand CD24's biological role, its potential as a biomarker, and how it can be targeted therapeutically. They focus on the mechanism by which CD24 engages with Siglec-10 on macrophages, leading to the suppression of phagocytosis and promoting tumor immune escape. The review highlights that CD24's primary role is to suppress macrophage-mediated phagocytosis through its interaction with Siglec-10 on these immune cells. This CD24 Siglec axis is a key mechanism by which tumors evade immune detection and destruction. Importantly, the study emphasizes that simply measuring CD24 expression isn't enough. Its functional engagement with Siglec-10 is a better indicator of therapeutic relevance. Therefore, any therapeutic strategy targeting CD24 must be guided by appropriate biomarkers and tailored to the specific tumor context. So, what does this mean for the clinic? CD24 represents a novel target within the innate immune system for solid tumors. However, its effectiveness is highly dependent on the tumor microenvironment and the presence of specific immune cells. To truly leverage CD24 as a therapeutic target, we need better biomarkers that assess its functional activity, not just its presence. Moving forward, the focus should be on developing standardized assays and designing precision immunotherapy strategies that combine CD24 blockade with other treatments to overcome resistance mechanisms. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.